You know, for the longest time, the way we've studied biology has been, well, by taking it apart. We grind up tissues to sequence their DNA and RNA, and more recently we've learned to separate them into millions of individual cells to get these incredibly detailed profiles. But in doing all that, we lose something absolutely essential: the architecture. Today we're going to talk about the technologies that are finally letting us put that biological puzzle back together.
So let's use a metaphor here, because it really helps. Picture this Lego brain as a piece of tissue. For years, bulk genomics was like throwing all those bricks into a blender and measuring the average color. You get a vague idea, sure, but you lose all the fine detail. Then came single-cell genomics, which was a massive leap forward. That's like carefully sorting all the bricks by their exact color and shape. Now you know precisely what pieces you're working with, which is amazing, but you've completely lost how they were originally put together. You've lost the context, the structure, the entire neighborhood where each cell lived.
And that, right there, is the crucial problem. A cell's function, what it actually does, is profoundly influenced by its neighbors, by its exact position, by the chemical signals in its immediate vicinity. You have to remember, biological processes don't happen in a vacuum. So when we disaggregate a tissue, we essentially erase all of that critical information.
Okay, so if taking the puzzle apart ruins the picture, how do we study the assembled puzzle itself? This is where a truly revolutionary new field comes into play.
And that field is spatial omics. Now, this isn't just one single technique. Think of it as a whole family of technologies, all united by one single powerful goal: to measure molecules, RNA, proteins, you name it, exactly where they are, right inside the tissue. This lets us do things we could only dream of before, like mapping how cells talk to each other, identifying these tiny specialized niches, and finally, understanding how the 3D layout of a tissue actually dictates its function.
Now, to actually achieve this, researchers have developed two major strategies. You can think of these as two different toolboxes, each with its own set of instruments designed for various specific jobs. Let's take a look at how they work.
On one hand, you have imaging-based methods. These are, very direct. They essentially use probes that light up, allowing us to literally see the molecules we care about through a microscope right there on the tissue slice. On the other hand, you have spatial barcoding methods. These are a little more indirect, but super clever. You place your tissue on a special slide that's covered in millions of unique molecular zip codes, or barcodes. Molecules from the tissue get tagged with the barcode of their specific location. Then, you sequence everything at once, and a computer uses those barcodes to reconstruct a complete map of where every molecule came from.
So let's dig a little deeper into that first toolbox: imaging. How in the world do you actually see dozens of different proteins or RNA molecules at the same time in the same tiny piece of tissue? Well, there are two really brilliant approaches to this.
All right, so the first approach, mass spectrometry imaging, it's incredibly cool because it sidesteps the limitations of light itself. Instead of fluorescent dyes, your antibodies are tagged with isotopes of rare, heavy metals. Then, a laser scans across the tissue, vaporizing it one tiny spot at a time. A mass spectrometer then analyzes that vapor cloud and can precisely identify which metals, and therefore which proteins, were in that exact spot. The result is an unbelievably clean signal with very little background noise. Now, the main constraint here is that there's a limited number of pure, distinct metals you can use, which currently caps the complexity at around 50 different proteins at once.
Okay, now the other major imaging approach is cyclic fluorescence. This method uses traditional fluorescent dyes, but it does it in this really clever iterative loop. So you stain for a few proteins, you take a beautiful high-resolution image, and then, and this is the key part, you chemically strip away or bleach that fluorescent signal. Then you come right back in with a new set of antibodies for different proteins, and you repeat the whole process, again and again. By stacking these images, you can build up a map of 100, maybe even more proteins. It's got a higher data generating capacity for sure, but the real technical hurdle is perfectly, and I mean perfectly, aligning every single one of those images from cycle to cycle.
So, with all these powerful tools on the table, a critical question comes up for any researcher who wants to get into this field, and it's a classic trade-off, one that really dictates the entire experimental design. How do you decide which toolbox to open? It all comes down to this: do you need to see every last detail in a very small area, or do you need a broader, more bird's-eye view of a larger landscape? And the answer, of course, depends entirely on the biological question you're trying to ask.
And here it is. This is the central trade-off in spatial omics today. Imaging methods are the absolute champions of resolution. They can see individual molecules. They can even tell you where they are inside a cell. Subcellular resolution. But all that amazing detail comes at the cost of speed and scale. On the flip side, spatial barcoding methods are built for throughput. You can analyze large tissue sections, you can process many samples in parallel, and you can get a view of the entire transcriptome. But the resolution is typically lower, often averaging the signal from a small group of cells rather than a single cell.
So let's make this concrete. When do you choose imaging? You pick imaging when your goal is deep profiling. Let's say you're trying to study a very rare type of immune cell and how it's interacting with cancer cells inside a tumor. For that, you need an imaging approach like mass cytometry. In fact, a landmark study did exactly this. They analyzed 500 breast cancer samples and discovered that specific spatial arrangements of immune cells, what they called TME motifs, were strongly correlated with whether a patient would survive. To see those critical life-or-death interactions, you absolutely have to have that subcellular resolution.
But what if you need to go broad instead of deep? What if your goal is to create a comprehensive atlas of an entire organ, like the mouse brain? Or maybe you're running a clinical study and you need to profile hundreds of tumor biopsies. In that case, spatial barcoding is your go-to. The ability to process many slides in parallel and generate that transcriptome-wide data makes these methods unbelievably powerful for large-scale discovery projects in fields like neuroscience and cancer biology.
So, even with all this amazing power, you have to understand we're just scratching the surface here. The field of spatial omics is moving at an absolutely breakneck pace, and it's all being driven by a very clear and incredibly ambitious ultimate goal. The Holy Grail. What is everyone in the field really chasing? It's getting the best of both worlds. It's the dream of combining the subcellular, pinpoint resolution of imaging with the transcriptome-wide, comprehensive depth of barcoding. Just imagine being able to map every single RNA molecule inside a single cell, for every single cell across an entire tissue, all while keeping it in its true, native three-dimensional structure. Closing that gap between resolution and profiling depth is the single biggest technological challenge that's driving all the innovation today. And that leaves us with a final really big thought. For the first time in history, we're building the tools to see biological systems not just as a list of parts, but as a fully assembled, functioning machine. As we finally start to rebuild this puzzle and map life in its true context, the real question isn't just what new things we'll find. It's what fundamental secrets of development, of health, and of disease are we about to uncover?